Wanted to go ahead and get started. Thank you, everyone, for joining us tonight for our uh, toasting for research. Um, I kind of wanted to get started and just introduce myself. I am Nick Beckham, and I am the president of the Alzheimer's of Central Alabama Junior Board. We are a junior board organization that is designed around doing events and raising money for research, as well as care, services, supplies, and awareness for those in our area that are experiencing dementia or have Alzheimer's in their family or caregivers. We're basically trying to assist in every way possible, both from awareness in the community, as well as assisting those caregivers with supplies they need, but support groups, and then of course our research funding, which we're here tonight to talk about. Just to give you a little bit of an idea of the junior board itself, for those of you that might be joining and that are interested, it's just a fun group. I like to say we're the fun side of the business. We're the fun side of the charity. We get to do a lot of events that you would attend anyway, and it's for great causes. You know, everything we do raises money, and it goes directly to people actually in our community. It doesn't get absorbed or sent elsewhere. It goes immediately to people that need it and people that are dealing with dementia in their lives. And it's something that's very important and personal to me. I work in skilled nursing, and I've done that for the last 13 years. And I work directly with Alzheimer's and multiple other types of dementia patients. How challenging this disease can be, and both in how it affects the individual that is afflicted with it, but also the families and the loved ones that are dealing with it and trying to care for those patients and those loved ones at their home. It's a very difficult process, and there are a lot of wonderful people all across the country and all across our state that are affected every single day by this. It's one of the number one issues facing our communities and facing our elder population. How I got started was I enjoyed going to charity events, and I met Vance Holder and Miller Pickett, and they were doing some fundraising events, and I loved it. I was all about it. I wanted to jump in with both feet and see if we could help out. And they had, at the time, an idea to start a junior board. Us and a small group of people thought that was a great idea. So we started, and we have been going for several years now, and it's been a wonderful thing. Our main fundraisers that we do are the Glow for the Cure golf tournament, which is an excellent event. I like to call it the best event of the year, is where we have the Highlands Golf Course and we do a glow in the dark golf tournament. So we have 9 holes of golf at twilight, which is the end of the day for non-golfers. And then we go into the clubhouse, we eat barbecue, we do some fundraising events, some updates, some good music, some prizes. And then as the sun sets, we go out and we have glow in the dark balls. The fairways are lit up with glow sticks. We try not to wreck the golf carts as best as we can, and we just have a blast. And so you can see the golf balls rocketing over the city. You can see the city skyline in the background, and it's just it's an amazing event. It's awesome, and it raises a ton of money for us. And the other event we do is the Ales for Alzheimer's, which is at Avondale Brewery or other breweries we can do, where essentially we have, again, live music. We have food, we get a proceed of sales, we do a silent auction, we do raffles, people come and enjoy the time outside with their families. We do a family-friendly event. Anybody can come, and it also does well for us. It's a great event, it's fun, everybody has a good time, and all of our funding we put towards the Alzheimer's of Central Alabama, and the majority of what we do at the junior board is fund the research program. So we're grateful to the sponsors that funded us this year. Unfortunately, we had to cancel, as many did, our largest events, the Glow for the Cure, as well as for Alzheimer's related to the COVID-19 pandemic, which is why we're all here on Zoom tonight. But we did have several sponsors listed, including Bradley Cooper, NHS Management, Comfort Care, Home Health and Hospice, Alabama's Best Home Care, Campbell Partners, Constangy Brooks Smith, Horton Lee. I see it's on the screen there now. Thank you, Miller. So that's a list of our sponsors, and we're very grateful to them, and they've always been huge sponsors of us with all of our events throughout the years that we've been running, and it's been a great thing. And since we didn't do it this year, we definitely had them as sponsors for tonight, and we're super grateful for them. Alzheimer's of Central Alabama, we have a long history of supporting research. Um, for those of you that don't know, we've actually funded 24 individual research grants, and that's been since 2001. That's a pretty amazing thing if you think about all the research that's done in a single grant. 21 of those were right here at the University of Alabama at Birmingham, which is a big deal for us because, one, it's local, but also because they're one of the largest research hospitals in the country, and it's very important to us to fund research. In 2018, we decided to take a small diversion in that, and the way we funded research with grants. You fund a grant, they do the research, and once the grant is over, that portion ceases. With the pre-doctoral program, which we started, we're actually funding a student. And we're hopeful that by doing this, we get really smart people, like the two we have with us tonight, more involved early in their careers with researching Alzheimer's and really putting and investing money into people that are going to spend their lives looking for cures and treatments for Alzheimer's. And we think that that could go even further as a much longer legacy than grants that we would do. So we're excited to hear from both of those scholars tonight, actually. Ashley Irwin is on with us, and Nawalzi Gwenza is on with us. But before we do that, I wanted to thank and introduce Tom and Susan Louder, who are here with us, who are longtime ACA supporters, and they do tremendous work to support not just the research that we do, but also the services and the support for families caring for loved ones with dementia. And tonight we're featuring their wine from Holman Ranch in Carmel Valley, California. I hope that's right.
You got it right.
Okay, and it's named Jarman wine, and that's very important because it honors Tom's first wife, Jarman, who had early onset dementia. So I'd love to introduce you guys and give you an opportunity to tell us about yourselves and a little bit about your story as it relates to your experience with Alzheimer's, and thank you for the help you're doing with us.
Well, thank you, Nick. Tom and I are very happy to share our wines. And it's just really, really impressed with Miller, who is a family friend, who has just also, her mother had Alzheimer's. So she is invested like we are, and it takes everybody, it takes a village. And we are really, really happy to be a part of that village. And I'm going to turn it over to my husband, Tom. And I hope you guys enjoy the wines. A big portion of our proceeds does go to Alzheimer's research, and we are committed to, our whole family is getting behind the care and the research for Alzheimer's.
Okay, thank you, Susan. I've been given the mission of making a toast, but I want to tell a story first, a personal story that would involve Jarman. And many of you may have heard a lesson that Buddha was teaching to his students about a man who was walking through the forest, and he turned around and he saw that an ambush or a pack of tigers was following him. So they started to chase him. They were going to eat him up. So the man, to escape, keep his life, he jumped over the end of a cliff, and he was hanging from this cliff only by a vine. And he looked up, and he saw the tigers, who were hungry and sniffing and were going to eat him up if he climbed up the vine that was holding him. So he was facing certain death if he climbed up, and he looked down, and if he let go of the vine, he would fall to his death. At that same time, as he's hanging with only the vine sustaining his life, he looked, as Buddha said, next to him, and growing out of the crevice of the cliff was this beautiful strawberry. And at that moment, he looked up and he looked down, and he looked at the beautiful strawberry. So he reached over and plucked the strawberry. And as Buddha said, how sweet it tasted. Well, 4 months before Jarman passed away, I was headed out the door to go to the office, and she was sitting with the caregiver, and she had not spoken to me with her Alzheimer's in at least a year, maybe close to 2 years. And our eyes met, and she looked up at me and she said, "I love you." So this toast that I'm going to make with the German wine to all of you is to all of you to sweet moments with your loved one in your life, as I had with Jarman at that very special moment. Okay, Nick, thank you so much. My hat's off to the junior board for inviting Susan and me in and letting us share our story and letting us share the Jarman wine with you. And with that, as I understand it, David is going to introduce himself, and we're going to talk about the wonderful program that is going on with that ACA is helping fund at UAB. Thanks so much.
Well, thank you, Tom, and thank you, Susan. Your contributions and your efforts and your donations are making a huge difference to people, and your story is something that really helps us remember and understand what we're doing. So thank you so much for that. And after now that they have shared their story, I'd like to introduce Dr. David Standard, who is the chairman of neurology and the John Whitaker Endowed Chair of the Department of Neurology. So I'm sorry, did I get all that right, Dr. Standard?
That's right. And thank you, Nick, for that introduction. And I want to thank Tom and Susan for hosting tonight too. And that was really a beautiful story, Tom. I hadn't heard that before, and it will stay with me. So thank you for that. So I'm really thrilled to have a chance to speak for a couple of minutes tonight on behalf of UAB and the UAB Department of Neurology and our partnership with Alzheimer's of Central Alabama and the Junior Board. I think this is, as you'll see, has been a very productive relationship, even though we've just begun with our first 2 pre-doctoral scholars. So I think it's a tremendous thing, and I'm really pleased to have a chance to say a few words about it. Alzheimer's could be the most important public health problem of the next 30 years. You know, there are 5 million people in the United States affected by Alzheimer's or dementia, and it's growing. It's growing because the population is aging. People are living longer, and the longer we live, the greater the chances of Alzheimer's. In Alabama, we expect that over the next 12 years, there'll be a 40% increase in people over the age of 65. This is the baby boomers of Alabama growing older. And as many of you know, people in Alabama tend to stay put. They don't leave. They like their home. They tend to stay in their home state, and we expect them to stay here and get older. And we expect to see more Alzheimer's in Alabama as a consequence of that. So that's a challenge that I think we have to rise to. We need to do better with Alzheimer's disease. We need to find treatments that slow the progression or cure the disease. This is really, I call it the pandemic that's not going away. We will get past coronavirus one day. I don't know when, but it will pass. Alzheimer's, unless we find better ways to treat it, will not pass on its own. And we are called to find a solution to this problem, or solutions. It may take more than one. So a couple things that are happening at UAB before I introduce our students just at a high level. On the one hand, we are actually engaged in a very broad redesign of our memory clinic because we've recognized for a long time that there are more people out there who need help than we can provide. It's a limited number of doctors that practice in this area. So together with the health system, the backing of Dean Vickers and Will Fernanny and President Watts, we've taken on a major redesign of our clinic. We're going to add a lot more advanced practice providers and a new approach to seeing patients with memory disorders. I can't go into all the details tonight, but I think over the next 6 months or a year, you'll see a pretty large-scale expansion of our ability to try and create ways for us to reach out to more people who are affected by Alzheimer's and memory disorders in Alabama. On the research side, we've invested heavily in that area. We've recruited some of the best and brightest from around the world, people like Eric Roberson, David Geldmacher, Jeremy Hershkowitz, Laura Volpicelli-Daly, Farrell Lubin, others that are really fantastic scientists and have built our capacity to study and solve the problem of Alzheimer's. And you know, the solution is coming. As many of you probably know, the very first treatment that could slow or prevent the development of Alzheimer's was submitted to the FDA for approval last week. Now, whether that treatment will get approved or not, I honestly have no idea. It may not. But in any case, it's only one of many. It's the first of a number to come. And I honestly believe, you know, within the next couple of years, we will have treatments that modify the course of Alzheimer's disease. Of course, that won't let up on the need for research. It only amplifies it. Once we have some success, we want more, and we want a treatment for everyone affected by this condition. So that's really where we're trying to go. The event tonight is really about the pre-doctoral scholar. So let me explain a little bit about what a pre-doctoral scholar is and why we call them that. So these are students. They are students that have completed their undergraduate work and have come to UAB to earn a PhD degree. These students are a critical part of our research enterprise. They bring not only some of the time and energy, but a lot of the best ideas have come from my students that have brought ideas to my lab. They have new thoughts and new voices and new views, and they provide a lot of energy to the environment. And then when they're done, they receive a PhD, they're a scientist, and as Nick said, they are probably someone who's going to spend the rest of their career working on Alzheimer's disease. So when Miller talked to me a couple of years ago about how could the Alzheimer's of Central Alabama be most impactful on the future, I said, well, research projects are good, but as you say, some work and some are not successful. They come and go. We do need to do them. We need to fund them. But if you want your dollars to make the biggest difference for the longest time, you invest in people. And if you invest in a young person who's bright and energetic and enthusiastic, and you convince them that they should put their energy into solving Alzheimer's disease, that's capturing the best minds for your problem. These students are all really smart. They could do a lot of things with their lives. There are many places they could go, but if we can encourage them to work on Alzheimer's, then they are likely to be part of the solution. They are the game changers of the future. I always tell people it's like planting a tree. If you plant a seed, it will grow into a tree. If you fund a pre-doctoral scholar, it will grow into an Alzheimer's scientist. That's where they come from, and it's so critical to have this support. So what the support from the Alzheimer's of Central Alabama does is lets us fund our pre-doctoral scholars for 1 or 2 years during their graduate training. This is important because the university does support them in their first year, but after that, it's really on the labs and the investigators to find money to pay their stipend and to enable them to work in the lab and work on their PhD. So we launched this, as you'll see, with Ashley Irwin, and then Nolzali is our second Alzheimer's of Central Alabama pre-doctoral scholar, and they're both doing fantastic work and really fueling the science, and I think fueling the future as well. So we're going to take questions at the end, so I won't go on too much longer, but I will go ahead and introduce Ashley, who is in her second year as a Alzheimer's of Central Alabama pre-doctoral scholar, and let her tell you a little bit about who she is and what she does.
Thank you, Dr. Standard. I'm going to share my screen really quick. I have a couple of slides that will hopefully make things a little clearer when I start talking about my research. Let's see. There we go. Can we see that?
Yep, looks good.
Okay, great. And thank you, Nick, and thank you, Miller, for inviting me and for funding my work. So my name is Ashley, and I am going to talk a little bit about I've been asked to talk about who I am and how I got involved in science and Alzheimer's research. And then I'm going to talk a little bit about my project, and hopefully everyone will stay awake. So a little bit about me. I'm originally from Georgia. I grew up in Middle Georgia. I've always been interested in science. I was the weird kid dissecting frogs and making magnetically powered turbines for my 3rd grade science fair, which nobody understood. but I ended up going to Georgia Institute of Technology. I got a degree in biology and Spanish there. And I studied these magnificent little creatures called rotifers. They're freshwater zooplankton. And anyone I've ever told that I study rotifers gives me the look you're all, I'm sure, making right now, like, what is that? But they're really fundamental to the ecosystem, and it really introduced me to what science is like and sort of the impacts that something so small can have. But I knew that that was probably not what I wanted to study long term. And so I decided to go to Georgia State University for a master's in neurobiology and behavior. And so then I started studying these furry fellows, and I looked at the neuroanatomy underlying social communication and recognizing important salient social cues. And that was just fascinating and really solidified my interest in neuroscience in general. I loved my experience there, but I think by the end I knew that, you know, social behavior wasn't exactly what I wanted to do. I'd always been fascinated with memory. And so when I decided to come to UAB, so I began in the medical scientist training program here at UAB in 2016. So what that means is that I am training for both an MD and a PhD. So eventually, someday, I will be a physician scientist, both a clinician and a researcher. And I brought with me my wonderful husband and my tiny terror of a 2-year-old. Well, now he's 2 years old. This is his first day of school a couple of weeks ago. They are really both my support and my motivation. So you asked a little bit about me. That's a little bit about me. And now I am in Dr. Farah Lubin's lab here, and we study this fantastic brain structure known as the hippocampus. So the hippocampus is really fundamental to the acquisition and formation and storage of our memories. And a couple of things led me here, but primarily when I think about who we are as people, without our memories, we kind of lose that component of who we are. And I wanted to understand how we form those memories and, you know, what happens when we lose them. And so Dr. Lubin's lab studies memory in both normal aging and in Alzheimer's, as well as in epilepsy, which can also have a memory component. So I really knew when I chose that lab that I would have an opportunity to explore memory formation, and then what happens in the brain when we lose our ability to remember. So I'm going to talk a little bit about the actual science. If the cellular component is not your thing, hang tight, because I'm going to talk a little bit about the behavior, about the actual memory in just a minute. So this is a figure that demonstrates what we call the central dogma of molecular biology. And it's the idea that our DNA gets translated into RNA, which is in turn translated into protein. And here I've positioned it at in the brain. So what I'm showing here is a synapse. That's where two neurons are talking to each other. This is what allows us to form memories. And ultimately, you have to have this transition from DNA to RNA to protein in order for the synapse to work. And in order for us to make new memories and to retrieve those memories later on, you have to have a functional synapse. You have to have this whole process of DNA to RNA to protein. And so my particular lab, Dr. Lubin's lab, is interested in a process that can alter how this expression of RNA can be changed, and that's called epigenetics. So what is epigenetics? Epigenetics is anything that changes the expression of RNA and thus changes the function of the cell without actually changing the DNA. So if I were more technologically savvy, I could have created a video where you put a ball right here at the top of this block, which I've labeled the Waddington epigenetic landscape. And the idea is that we all start with the same DNA. And then as we come down the ramp, we get shifted into different slots based on what genes are being expressed. So what makes a brain cell different from a kidney cell, different from, you know, a retinal cell, is dependent on which genes are expressed. And something that drives this differential gene expression is epigenetics. Now, there are a very wide variety of different epigenetic marks, but one that I'm particularly interested in is the regulation of this by something called long non-coding RNA. And one particular long non-coding RNA, and that would be called neat1. It's very neat. So back when I was showing the DNA to RNA, all of that is describing something called messenger RNA. It becomes a protein, but there's actually a lot more RNA in your cells that doesn't become a protein. It doesn't code for something specific. And originally we thought this was junk RNA, you know, and the reality is we're learning more and more that I promise I'm not going to talk through all of these different pathways. This is just to show you how complex and how integral these long non-coding RNAs can be to the function of the cell. This is just one example, and the one in particular I'm interested in. And for those of you who might actually be trying to figure out what's going on, there are quite a few pathways that we already know are involved in the Alzheimer's phenotype. So A-beta, endocytosis, clearance of different plaques, things like that. So what do we know about neat1 other than it's very neat? Well, we know it's expressed throughout the body. It has relatively low expression in the brain, but it is there. And some work from a previous grad student in my lab has shown that in aging animals, mice in particular, you have this elevation of neat1 expression, particularly in the brain. So while it's normally very low, as these animals get older and older, the neat1 expression is increased. Now, I'm not going to show you all of his work because he did an amazing job, but he also showed that if you can decrease that neat1 in these old animals, you can improve their memory. So when I joined the lab and I was interested in memory and particularly in Alzheimer's, I began to ask questions about, well, what is neat1 doing in the context of Alzheimer's disease? So to answer this question, I'm using an Alzheimer's disease mouse model. And it has, it's a transgenic mouse model that has two different familial Alzheimer's genes. So most of Alzheimer's is sporadic. There are a lot of different factors that contribute to the onset, but there are several different families where there's a known genetic component, and this mouse has two of those different mutations. And so the benefit is that these animals begin to show similar characteristics at a very young age. So roughly 3 to 4 months old, we start to see cognitive impairments, amyloid plaques, and then you see things like gliosis, which I'm only mentioning now because I'm going to talk about it in my very last slide. And so gliosis is abnormal numbers or activity of a specific cell type called glial cells or astrocytes. So this is the mouse model I use, and you might be wondering, how do you test memory in a mouse? And yes, we use mazes. I have used mazes. I'm sorry, I'm not going to show you my maze data today. What I'm going to talk about instead is something called context fear conditioning. So I mentioned early on that we study a brain region called the hippocampus. So one of the things the hippocampus does very well is help memories that are involved in context. So if you lose your car in the parking lot, you know, we're talking about a memory related to where you are in space. So in order to test these animals, we place them in a novel context, something that is different from their home cage, and we allow them to explore, and then we deliver a shock. And I just want to say up front that I am not in the business of harming mice. I have tested this shock on myself, both on purpose and sometimes not on purpose. It is startling more than painful. But what it does is it encourages the mice to remember that context. And so when you return them to that novel space, or what was once a novel space, they're afraid. They're concerned that they're going to be shocked again. And so in a mouse, the reaction is to freeze. That is a fear response in a mouse. So what we can do is then test these animals and see how much are they freezing. So in a normal young mouse, you know, the equivalent of a young person who remembers where they left their vehicle, they are going to freeze, 40 to 50% of the time in their environment when they're returned to it. But in an Alzheimer's mouse, you see significantly less freezing behavior. And so many people interpret this as being a failure to remember the context. So there are lots of other ways to measure memory, many of which I'm doing, but for today, I'm just going to talk about this one. Because over the summer, I was very interested in seeing what might happen if I repeated the experiments that my lab mate had done in aged animals. So where he decreased the amount of NEAT1 in the brain, in the hippocampus specifically, and then tested these aged animals' memory. I wanted to know, okay, if I decrease NEAT1 in an Alzheimer's mouse and then test their memory, what happens? So I'm going to walk you through this figure. Here on this axis, we have freezing behavior. So the blue bar is our wild type animals. These are normal animals who do not have the Alzheimer's gene. They're young. These are 4 to 6 month old animals, which is, young to middle-aged in terms of mice. And they have an appropriate level of freezing behavior when we return them to that context. So these are animals that are introduced to a novel cage, they're shocked, and then 24 hours later, we return them. So then, down at the bottom in this purple bar, these are Alzheimer's animals that received basically what we would call a sham injection. They received an injection in the hippocampus, but there wasn't anything in the injection, just saline. And so in theory, there should be no modification of their memory. And what you can see is, as we would expect, they're not freezing as much. So we might interpret that as, well, maybe they don't remember that context as well, which is what we would expect from an Alzheimer's animal. And then in my final group here in the green bar, I wanted to know what would happen if I just knocked those NEAT1 levels down, hopefully making them similar to what you would see in a normal control animal. And as you can see in the green bar, we have a pretty significant increase in the amount of freezing when we return them to this novel context. And so this is some pretty promising preliminary data, at least for my project, that shows that NEAT1 is capable of mediating memory, at least memory that's taking place in the hippocampus in these Alzheimer's animals. So my future work going forward is I want to know how this is functioning, or how this is happening. How is NEAT1 controlling memory in the context of an Alzheimer's model? And so one of the primary components of my future work is looking at it in a cell type specific fashion. So for people who don't do neuroscience research every day, you probably don't think about the fact that the brain is not a homogeneous structure. There are all kinds of different cells in your brain. And I showed you an example of a synapse, so two neurons talking to each other. And really neurons are the ones who get most of the attention in the brain, and for good reason. They're very important cells, but I have, a soft spot in my heart for the underdog. And so I'm interested in this cell type, these glial cells called astrocytes. And we already know that with both normal aging and then with mild cognitive impairment that can often progress to Alzheimer's disease, you see a change in how these astrocytes are functioning. And so I'm particularly interested in how NEAT1 is acting to affect the function of astrocytes. And this is important because when you're thinking about developing treatments for something like Alzheimer's disease, you want to be as specific as possible. My experiment that I showed earlier with the NEAT1 suppression and the context fear conditioning is like hitting something with a sledgehammer. I just knocked it out of every cell. And there are a lot of different side effects that may occur. NEAT1 is obviously playing an important role in the function of these cells. So I want to know exactly where it's having its greatest impact in the context of Alzheimer's disease so that then we can target it very specifically. So that is just a little short blurb about my research right now. I'd like to thank all the people in my lab and my committee, including Dr. Standard, who've helped me with this project, and my program, and then of course the ACA Predoctoral Fellowship. And please, if anyone has any questions.
All right. Well, I think we're going to try and take questions at the end from all three of us. So we'll go ahead and let Nawazi talk a little bit about her work. So she's the newest Alzheimer's of Central Alabama predoctoral scholar, just starting in with this program. And I'll let her go ahead and tell you a little bit about herself and her work. I think you're still muted. There you go.
Sorry, thank you. Thank you for that introduction. I'm going to try and share my screen now. Sorry, a lot happening on my screen right now. Okay, I hope everyone can see that.
That looks good.
Thank you very much. Okay, awesome. So I wasn't very sure about what the tone of this evening was going to be. So I just decided to keep it very light and really just tell the story about how a girl from Africa came to find herself in the deep South. My name is Nolwazi Twensa. I am from South Africa, a very aptly named country in the southernmost part of Africa. Now, I am a little ways away from home, so I thought I'd tell you a little bit about how I got here. From as early as I can remember, I have been interested in the pursuit of knowledge. I've envisioned myself in a white lab coat since I was 10 years old. In fact, in my native language, my name means mother of knowledge. I think my mother knew to plant that seed early on and set her hopes for me quite high. As a kid, I was always asking, but why? There wasn't a piece of information I found too trivial or a point of curiosity I could leave unexplored. It drove my mother crazy, which in all honesty, I don't even feel that bad about, given that she was the one who wished it upon me with my name. This curiosity sort of never went away, and eventually I got to the point where I realized that some of the questions I was asking didn't have answers yet. Well, I needed those answers, so I decided I was going to go find them myself. So off I went to university to study in the field I thought would have all the answers for me, science. I walked into the university completely doe-eyed. All I knew is I wanted answers. I thought to myself, chemistry would help me understand how the world works, and neuroscience would help me understand how we work. Turns out my university didn't offer neuroscience until an honors level, so I just went with geology. And for a minute there, I really thought I could be a geological chemist. Absolutely false. Anybody who knows me knows I am a complete city girl, and something about leaving the comforts of home to go camping and hiking just to look at rocks just didn't sit right with me. So I changed directions completely, decided to study the ocean, and stumbled out of my bachelor's with a BSc in chemistry and ocean and atmosphere. I got to the end of my undergraduate degree, and honestly, I had more questions than answers. Anybody who tells you that scientists have all the answers for you is telling bold-faced lies. As I get older, I'm learning that science is just a bunch of people fishing around in murky waters and hoping to pull something out that makes sense. But that lesson really didn't dawn on me until I got into my postgraduate studies. At this point, I had a choice to make. I could go back to my original goal and finally spend time pursuing neuroscience, or see how far chemistry could take me. I was still hungry to understand all that I could, and I didn't yet feel I'd learned everything from chemistry. So I decided to follow my curiosity and do my honors in chemistry. Almost in my first week of my honors degree, I learned about these fabulous structures called metal organic frameworks and fell madly in love. These impossible salt-like crystals seemed to have endless possibilities, and in my eyes, could fix all the world's energy problems. So my curiosity took me deeper down the rabbit hole, and I went on to complete my master's in chemistry, specializing in the study of metal organic frameworks. As I was closing out my master's studies, questions around my next steps started coming up more and more often. Would I be continuing to do my PhD in chemistry? Would I be starting my career in industry? For someone with my credentials, a career in field chemistry seemed like a natural next step. I didn't know how to answer any of these questions, but I did know I was getting exhausted of pursuing what I was starting to see as very theoretical knowledge, and I'm starting to think about what I really wanted my impact and legacy to be. It was around this time that I had some serendipitous encounters. First, with a few of my students who asked a seemingly innocuous question, why did you study chemistry and oceanography? To which I gave a seemingly innocuous answer. Well, I wanted to study chemistry and neuroscience, and the university didn't offer neuroscience until honors, so I did oceanography instead. To which they responded, well, why not study chemistry now? To which I said, huh. Of course, I'd had similar conversations and considerations before this, but this was the first time this question coincided with my desire to tackle more human problems. And I thought, why not apply my analytical chemistry brain to solving problems that could affect us all one day? Aging. The cogs were turning, but it wasn't until a second serendipitous encounter that things really started moving. A friend had come back home to visit and we decided we wanted to watch a movie. We'd gotten there early and gone to chatting while before the trailer started. She told me about a program she was a part of that helped her pursue her studies in the States, and she thought I would be a good candidate for it too. At this point, I'd never heard of a Fulbright, but she sold the program so well, I thought I'd give it a chance. I got into the applications, and I'm sure in some shape or form, everyone has gone through the hellish experience of applications. The Fulbright application is particularly tough with layers of essays, interviews, tests. I really had to sit down and look at my life and what I wanted from it to have a chance of convincing someone else to invest in my goals. And thank goodness I did, because it set me here. Suddenly, I was in the deep south. To say I felt like a fish out of water would be grossly underselling it. I hadn't done biology since learning the mitochondria was the powerhouse of the cell in high school. And suddenly people were asking me what research I would be dedicating my life to for the next 5 years. I was lost and in over my head when all I knew is I wanted to study Alzheimer's disease. But then I went to an interview with Dr. Laura Volpicelli-Daly, who would eventually become my PhD supervisor. And she told me about a protein in the brain that acts like a crystal. Suddenly I found a place where my understanding of crystals could be used to make things better for everyday people, all because of a protein that acts like a crystal and in doing so can contribute to the psychiatric symptoms and cognitive decline associated with Alzheimer's disease, and that further isolates people living with Alzheimer's disease from their family and loved ones. I would learn later on that a lot of proteins act like crystals, but at the time, this felt like serendipity at work for me yet again. So I joined a lab 8,558 miles from home, studying Parkinson's disease, and ultimately took on a project in Alzheimer's disease anyway. Then I tried my luck with another application for the ACA Pre-Doctoral Scholars Program and somehow ended up talking to you today. So what exactly am I doing now that I'm here? Well, I mentioned earlier that I was drawn to this lab by this crystal-like protein, but what exactly does that mean? Well, some of you may have done this really cool experiment where you kind of have a colored solution of salt and you dangle a string in that solution and let it sit for a while. When you come back, you have a big salt crystal dangling on a string. It's a really cool experiment, but I'm sure you can imagine it's not exactly desirable inside a living cell. This is not a perfect analogy, but it does demonstrate this cool principle of crystals growing into a larger mass. Alpha-synuclein is a protein that sort of behaves like this. In normal cells, when alpha-synuclein is behaving as it should, these masses get cleared out of the cell and don't cause too much damage to the cell. Sometimes, though, the alpha-synuclein doesn't behave correctly and starts to form these big masses that grow and grow, sort of like the salt growing on a string, until the cell gets very sick and dies. This is sort of what we think is happening in some of the brains of people living with Alzheimer's disease. And what we want to do is to try and understand what is initiating this process so that we can eventually find a way to stop it or even prevent it from ever starting. Truth be told, my research probably won't produce a cure for Alzheimer's disease. I know it's disheartening. And honestly, it's been a very tough realization for me too. But I'm coming to see that that's just not how science works. The saying, Rome wasn't built in a day, could not be more apt than in the work of research. A more realistic goal is that the work that I do inspires a generation of smarter, luckier scientists who build on what I know and build on what those who came before me knew until we finally find an answer that brings hope back for all the people that this disease has affected. More realistically, my work will form a basis of knowledge because before we can even start guessing at a cure, we have to understand the proteins that make up the system that could possibly go wrong and maybe even cause disease. Because the crux of the problem here is that we're trying to cure disease when we don't even understand how it starts. And that's the answer my research is trying to get at. And that's really what I want to do with my life. I want to inspire better scientists and encourage smarter minds to put their efforts into solving this puzzle through my work now as a researcher and later as a scientific investigator, a mentor, a teacher, and eventually a policymaker, so that everyone has access to the information we create now, as well as access to the resources that can encourage them to put their efforts into the work until finally someone somewhere has a cure. So thank you for investing in my work and the work of great scientists so that we can all work together to find a cure. Thank you.
All right, well, thank you for that great presentation. And I wouldn't sell yourself short on finding the cure. You know, one of the things about science is sometimes you don't understand how all the pieces fit together until suddenly the puzzle is solved. So
thank you. I hope so too.
All right. Should we bring back Ashley? And I think we have time for some questions. Do you want to moderate Nick, or is Nick still out there? Yeah, I think we could take questions from the Q&A or the chat box, or how do you want to do this?
Sure. Let me check the chat box. If anybody has any questions, you can just type them in the chat box, and I can read them, and that'd be fine. I will ask one question. You know, just based on my work, and I'm not sure how much you can answer, or I can give you some of my own, but what I've seen is a lot of influence from this pandemic on our Alzheimer's population. Mine's mainly related to the social factors, being the decrease in interaction with individuals that they know, the isolation, those type of things. And I've seen heavy declines in that, unfortunately, and we all have as an industry, and we're trying to figure out ways that we can support them via Zoom, those type of things. But just as far as maybe chemically, for our scientists that are on here, is there anything that you guys have seen from a mental aspect that this COVID-19 virus has impact, or is that just way too early to know anything like that?
Well, we can all take a shot at that, I guess. I mean, I would certainly say the social aspects are very important, right? I mean, these are, you know, Alzheimer's, the social interactions, the people around you are critically important, and this virus is certainly taking those away. From a biology perspective, the question of will this virus promote brain diseases years off in the future, I think that's what we all worry about a little bit, to be honest. That you know, there is a history here. The influenza pandemic of 1918 led to a rash of Parkinson's disease cases. And so, and there are really striking inflammatory features to this virus. And so we don't know, but I think everyone's a little concerned. And the sooner this pandemic is over with, the better off we'll all be, no doubt about that part. But I don't know if Ashley or Nawazi wants to, I'm sorry, I'll get your name right eventually. Nawazi wants to comment on that.
Yeah, I don't really have a whole lot to add. And I think the problem is that nobody really knows. Nobody, I mean, I'm sure there are labs who are actively working on it, but we won't know for a while, right?
Yeah.
I honestly, I wouldn't hazard a guess at what COVID could possibly imply for Alzheimer's disease. I'd be very interested, though, to see if there's something that we see and like noticeably see a difference in the long term. So I'll be keeping my eye out on what the research says.
Yeah, I think a challenging part of it will be that, you know, in order to do proper research on it, particularly from a chemical standpoint, you'll need lots of participants that are still living well after the fact of recovering. And I'm not sure that the time will be allowed, because obviously the focus now is to stop the spread and versus that. But I do think this virus is going to be around. So I do think
that's true. Although once the immediate emergency is passed, we have a long list of people we know had this virus. We've tested, you know, tens of thousands, hundreds of thousands at UAB, and we know who all of them are. So it's actually going to be possible, you know, 5 years from now to look back and ask some of these questions.
And I do see one question already in the chat box from Robert Baldwin asking if you could comment on the new drug that you mentioned that was pending FDA approval and expand on that.
Yeah, so the drug that was submitted was a drug called aducanumab from Biogen. And this is a drug that works on the amyloid protein. So the amyloid protein is one of the crystal proteins that builds up in Alzheimer's disease. And aducanumab is intended to remove that protein from the brain. And it actually does do that. It was shown to do that through imaging tests. And then the question is, well, does that help people any? And the clinical trials, essentially there's one trial that looks pretty good, another trial that maybe doesn't look so convincing, but they took that to the FDA and presented that as evidence that this wasn't an effective treatment. The FDA, if you're watching the stock, I don't own any Biogen stock because that would create too many problems for me. But Biogen stock went up 40% in one day after the first presentation, and then it went back down 40% the next day. So I don't think even Wall Street knows what to make of this. I think the data is a little bit mixed. There's a signal there, but it's not as strong as we'd like. I don't know if aducanumab will get approved or not, but there are others in the pipeline, and I think it's only a matter of a relatively short time before one or more of these makes it through. So I don't know if you guys have been following this or want to comment on this.
Actually, I kind of have a question myself. So, I mean, as I mentioned in my talk, my background is mostly in chemistry, and if you're comparing kind of the I guess, the timeline of like chemistry to biology, it's a very long time between like doing something like fiddling with making a drug to putting it in a human being. Mm-hmm. So I guess I'm kind of just curious, like, what is the FDA approval process? Like once you've kind of proven this in whatever models, then how long does it take thereafter to finally actually get into people?
Well, from the approval to into people doesn't have to take that long. The FDA standard is, safe and effective. That's their standard. Now, doing the trials, these trials have been going on for maybe 5 or 6 years with this particular, it's an antibody treatment. So it's an antibody to amyloid, and it removes amyloid from the brain. So they've spent 5 or 6 years in clinical trials getting to this point. Once it's approved, it can come to people pretty quickly. But it's that long period of 5, sometimes 10 years between an idea, getting it to the data, and getting to present to the FDA. That's really the big lag, and many drugs never make it. So there is a sort of a funnel effect here where many ideas are tried and only a few ever get even to this stage.
There was one more question that I see asking regarding Alzheimer's disease. Is it the still most prevalent type of dementia? That says, I've heard that 60 to 80% of cases are Alzheimer's disease. I also hear more about Lewy body, FTD, et cetera, and wondered if the percentage has changed at all.
Well, the percentage hasn't changed. The most common dementia is Alzheimer's, no question about that. Lewy body, and Nwazi may want to talk about Lewy body a little bit, is a related kind of dementia. It's a little bit different. And then there are also sometimes dementia from other causes like vascular disease. Strokes can cause dementia as well. But Alzheimer's is by far the most common still. Some people have more than one kind of pathology in the brain too. That's the other thing. We talk about these as if they're completely separate, but when you actually look at the brain, sometimes you see Alzheimer's changes and Lewy bodies and stroke, and all of these things are there. So.
I don't know. I don't know how much more I can add to that. I mean, yes, Alzheimer's disease is in fact the most prevalent neurodegenerative disease still. I know that Parkinson's disease is, I think, a second to that. Not a very close second, but I think Parkinson's disease is second, which is a type of, and falls under the umbrella of Lewy body dementias. So yeah, I think if that answers your stats question at least.
Sure. And then the let's see, other question that someone sent me was if you could comment, Dr. Standard, on the funding for, let me make sure I get this right, for the new Alzheimer's Dementia Research Center.
Yeah, so this has been a long sought goal. So Dr. Roberson has been awarded a Alzheimer's Disease Exploratory Center by the NIH. So these are centers which are really the anchor for Alzheimer's research. And we actually had one. Lindy Harrell had an Alzheimer's Research Center. And when I first arrived at UAB in 2006, that was still active. But that center expired in about 2000 or 2007, 2008. So we've spent the last 12 years trying to get that refunded. So it's a kind of a major victory. What an Alzheimer's Research Center does is it facilitates research on people with Alzheimer's or people who are at risk of developing Alzheimer's. So it recruits people, does testing for memory, does things like PET scans where you can actually look for the Alzheimer's protein in the brain, and follows those people over time. And it makes it possible to do many different kinds of studies in that group of patients. So it's a group of people who volunteered and have been really studied very thoroughly at the beginning. And now if we have a new drug we want to test, those would be the people to go to because we know everything about them already. And it makes a kind of a ready-made group that's volunteered and is ready to go, and really accelerates the research tremendously. So it's kind of the cornerstone of many different aspects of Alzheimer's research. And so that was just refunded this summer. We're really excited about that. And that was done through a combination, we got there through a combination of university and community support. We essentially had to build and operate an Alzheimer's center using support from the community and the university and the institution to get to where the NIH would fund it. Now they funded it, and we hope to grow that and carry that forward really as a cornerstone of much larger research programs all around Alzheimer's disease.
That's great. Well, I'm looking and I'm seeing if anybody else has any questions. Someone just said thank you so much, and I would like to say thank you to everybody, to all of our scientists here today, and also to Tom and Susan again for your continued support for the brilliant minds that we have here today, and to Miller for continuing her efforts with Alzheimer's of Central Alabama. There is, I can tell you guys from personal experience, there are thousands of lives that this touches, but that you touch, that everything that you do to support this cause, to give support to people that are researching this cause, all of those things are making a difference. It's making an impact. Just to emphasize what Dr. Standard said earlier, I think this is the issue of our time. I see it every day in what I do, and it grows and grows and affects everyone. I think each and every person on here knows that anyone you talk to in your social circle is in some way affected by Alzheimer's and dementia, and that should tell you exactly how prevalent it is and exactly how important it is that we both continue to support those that are having to deal with it now, and also to continue to look for a cure and look for a treatment and continue this research. So thank you so much to everyone here. And we look forward to doing more events in person at the end of this pandemic where we can all drink in person. Oh, and with that, it is a toast to research. So I'm going to raise a glass for those of us. I know Tom and Susan are participating. I'm sure everyone else is. Didn't want to be on camera.
All right.
Thank you very much, everyone.
Listen, everybody, this is Miller Piggott, and I want to thank Nick and of course Dr. Standard for helping moderate this, and Ashley and Nawalzi, and I hope that you can all see and appreciate why we're so very proud of our junior board and the work that they're doing. I think that it is the cornerstone of what ACA does, is helping families keep their loved one at home. But supporting research is another really, really important part of our mission. And we're proud to be able to provide this for the university. And of course, we're excited to see where your two careers go, and would like to continue to follow with you. And Tom and Susan, y'all have just supported us so graciously for so many years. And thank you so much for your help tonight, and the support with the wine too. We appreciate that.
Our pleasure.
All right. Well, thank you all for the time this evening.
Good. Thanks, everybody.